Question 4. This shows the fluid mosaic model. Of the cell membrane. Alright, label X as the protein. And remember, actually, X is different from the protein here, which is a channel protein. You see the channel. Now I showed it in blue. They are not the same. So you better label this as a protein. And then when you label this, you either label it as phospholipid, and you should not write down water-loving head, because this is not a name of the molecule. It is just a description of the structure. Then look at the question B. It is given that pigment will be released from the cell if the membrane are damaged. So the student Gary places identical beetroot tissue. That is, they prepare some tissue of the same size already. Same size in each test tube. Identical. And then each tube contains same volume of alcohol. However, it is different concentration. So therefore, everything are the same. Everything is the same. The beetroot size are the same. The volume of them are the same. And the only thing that you are talking about different is the concentration. So this must be the independent variable. And then they look for the color differences, the intensity. So control variable would be those that are kept the same. And you can mention about the size of the beetroot. And also, you can name about the volume of the alcohol. And you can also mention about the duration of immersion. That is how much time you keep the tissue in the alcohol. All these will affect the rate of diffusion. And of course, some of you also mentioned about temperature. Temperature is also a possible way that will affect. So temperature. And then now let's move on. And this question asks about independent variable. That is the cause of the change. And therefore it is the concentration of alcohol. When we change different concentration, it is causing the result of different light intensity. So from the result, we see that D is highest in color intensity. That means it shows that most pigment leak out at D. Why are there most of it? Actually, if the alcohol has higher concentration, it will cause most damage to the membrane. That is, more phospholipid is dissolved, and the membrane will become more permeable. Therefore, it will lead to a faster diffusion of pigment. Why? More phospholipid is damaged or dissolved, then there will be more places for the pigment to come out or leak out. For part 4, after 3 hours of time, remember it is 3 hours, so it is quite a long time. he found that the color become the same. All of them are the same. That is, all of them become the same color. It's just an explanation. If we assume that all the beetroot tissue are the same, then they should have the same amount of pigment. If they have same amount of pigment, after a long hour, 3 hours, all of the pigment will inside available, that is, will leak out. That is, all pigment, the same amount of pigment leak out to the solution. So they have the same color intensity in the end. So for question 5, when you look at the diagram, one of it come from a plant, the other come from human. And if you have no idea of what they are, let's take a look back. Actually, all right, the tissue here come from the stem of a plant. Here is the stem, and you see, when we enlarge the stem, we will have the structure. And then this structure, when we enlarge it, we will see something like this. And when you enlarge it, you will see something like this. So you see, they are different. And these are the big holes, which are the xylem. And the phloem is there. And then if you enlarge this part, you will see something like this. And therefore they are the xylem of a plant stem. And on the right, we have the vessel of a human, and vessel P compared to this. So you see the differences in the wall. So here, P has a very thick wall. All right. And this side has a thinner wall. So actually, we can confirm that this is an artery of human. When we look at the question B, so you see how. How means asking for method and mechanism. The substances inside can be maintained. That is how the transport, that is how substances inside are transported. Asking for a reason, asking for a method, not a structure. Therefore, you have to answer a method of movement inside. Therefore, in the xylem, the content is moved by transpiration pull. And in vessel P, the artery, the blood inside is caused by pumping of the heart. And you should not only write heart, all right? You must write pumping action of the heart. Why? Pumping is the method. Then part C, it is asking about the structural adaptation. And you are requested to answer according to the diagram only. That is, you answer what you see. And in the diagram, you will see here, very thick wall. All of them have thick wall here. And thick wall in P. So the first answer is thick wall. Alright? And you have to explain the common. That is, it is available for both structure, common structure. And how they are adapted to the function. Therefore, it is the mechanical answer again. The thick wall provides mechanical support so that it can prevent this from collapsing during transpiration. And for a artery, it can withstand high blood pressure. Don't say prevent collapse. Okay, it can withstand. It can withstand, not provide. The second part would be two of them as seen in the diagram, a hollow tube, so that the substances inside can pass through with little resistance. Question 6. It talks about rate of secretion of two hormones at the pancreas at different blood glucose level. So let's look at the drawing. Here is the glucose level, and here talks about hormone A, and on the right we have hormone B. So you see hormone B, the change is like this. Alright, and for hormone A, it is like this. Question A, which hormone play an active role in control of blood glucose level when the level drops from normal to 40? Normal here, and then it drops from 40. So we look at the changes on this side only. What happened? The glucose level that is the change from there to there. So when the glucose level drops from normal, that is around 90 to 40, what happened to the hormone there? Hormone A, the concentration goes up. And for hormone B, what happened to hormone B? It just remains at very low level, no changes at all. So which is responding to the glucose level? And of course, we would say A. So in the answer, then we say when glucose level drops from 90 to 40, hormone A increases in concentration. Hormone A increases in concentration, and hormone B remains unchanged at very low level here. Therefore, we say hormone A is the hormone that can control. And then for name hormone A. So what is the hormone? Hormone A helps control when the glucose level is very low. So which hormone? And therefore your answer would be glucagon, which you talk about how that means more glucagon. When glucose level is very low. Alright, so what is the mechanism? How? So it is more produced, it will stimulate the liver. Remember, stimulate the liver to convert glycogen to glucose, so that more glucose will be released and the glucose can return to a normal level. So for the last essay question, we can look at the structure like this. For placenta, lungs, and small intestine, all of them have similar situation because they are places for diffusion and exchange. They have a surface area which are large for diffusion. They must increase the concentration gradient, and they must shorten or lower the diffusion distance. So you mention the structure one by one, and you must state them clearly. For example, air sac, you must say the air sac are highly folded. And in placenta, you mention about the embryonic membrane form finger-like, and just like V-like, you must say finger-like. So look at the structure and you will have write it out as usual.